In our last video, the question I left you with was about the differences between linear electron flow and cyclic electron flow. And so hopefully you noticed that in linear electron flow, there were two photosystems, photosystem 2 and photosystem 1. In cyclic electron flow, there's only one photosystem and the electrons cycle. So there's no water needed to split over here to replace electrons lost because all these electrons just continually cycle. So water's not needed as an input, and also there's no NADPH as an output. So there's the final electron acceptor is going to be this chlorophyll, so it's just a cycle, and so there's no initial water splitting to provide electrons, and there's no final electron acceptor. So the only product of the cyclic electron flow is ATP. So light energy is used to create an electrochemical gradient that's used to make ATP. So why would plants want to do this if they're only getting ATP out of it and they're not getting NADPH? Because we learned that both ATP and NADPH are needed in the Calvin cycle. But you'll notice that the ATP and NADPH needs are not balanced in the Calvin cycle. There's actually more ATP needed over here. And so plants actually run a little bit of the cyclic electron cycle all the time along with the non-cyclic or the linear cycle, and that helps them make a little bit of extra ATP that they need to balance out the numbers. So that's an alternative pathway for the light reactions. Let's look at alternative pathways for the Calvin cycle. So there are two pathways that some plants run in addition to the Calvin cycle. The Calvin cycle has as its first intermediate, if we go back here, the first stable intermediate in the Calvin cycle is a 3-carbon molecule, this phosphoglycerate. So because of that, the Calvin cycle is known as a C3 cycle. But some plants, in addition to that C3 cycle, also have a C4 pathway or a pathway called CAM. And why is this necessary? In order to understand this, why plants need alternative pathways in addition to the C3 Calvin cycle, it's important for us to understand the enzyme, the main enzyme that's responsible for carbon fixation. And that enzyme is called Rubisco, which is short for ribulose bisphosphate carboxylase oxygenase. And now you know why we just call it Rubisco. Much easier. Now, enzyme names are all often, I won't say always, but often very descriptive. So, for example, this enzyme, we notice that it's an enzyme because it has this ASE ending. And there's actually two ASE endings. It's carboxylase oxygenase. And you'll notice that the beginning of the enzyme starts with the name of the substrate it acts on. So it's called ribulose bisphosphate, and that is because the substrate that it acts upon, and we can see this in the first phase of the Calvin cycle, Rubisco is this enzyme here that catalyzes the addition of a carbon dioxide to ribulose bisphosphate. So this first part of the name, ribulose bisphosphate carboxylase, shows us that one of the main functions of this enzyme is to carboxylate or add a carbon dioxide to ribulose bisphosphate, RUBP. But notice that this enzyme also has oxygenase activity. And so that means that sometimes instead of adding a CO2 to RUBP, sometimes instead Rubisco will add an oxygen. It will oxygenate RUBP. So In the Calvin cycle, what typically happens is RUBP, this 5-carbon molecule, will get carboxylated by adding on a CO2, and that makes 3-phosphoglycerate. And these then are, this is the fixation step, and then in the reduction step, ATP and NADPH are used to reduce the phosphoglycerate to G3P, and then the G3P, once we get 6 of them, we can use that to make a glucose. Or sorry, if we get 2 G3Ps, we can use that to make a glucose. But for every 12 CO2s, no, let me, sorry, for every 6 CO2s that are fixed, we're going to get 12 phosphoglycerates and 12 G3Ps, and 10 of those G3Ps will be recycled back to RUBP, and 2 of those will be used to make a glucose. So this is the way it happens in the Calvin cycle. But if Rubisco, instead of carboxylating RUBP, if it instead oxygenates RUBP, then instead of getting 2 phosphoglycerates, we get 1 phosphoglycerate and 1 2-phosphoglycolate, so a 2-carbon molecule. So if we add an oxygen, instead of getting 2 3-carbon molecules, we get 1 3-carbon and 1 2-carbon molecules. So this is actually disadvantageous to the plant. So from what you've read, what is the main disadvantage of plants doing photorespiration? Well, the point of the Calvin cycle is to fix carbon dioxide so that we can make glucose from the G3Ps. But if oxygenation happens and oxygen is added to the ribulose bisphosphate, then we get these 2-carbon molecules, the phosphoglycolates. We get a 2-carbon molecule and 1 3-carbon molecule. But in order to make glucose, we need 2 of these 3-carbon molecules, and so this 2-carbon molecule has to be converted into a 3-carbon molecule. In order to do that, you have to have 2 of these 2-carbon molecules. You have to go through this complicated pathway, which actually strips a carbon off. So we're kind of going backwards. We really want to incorporate carbon dioxide into ribulose bisphosphate, but this process is considered to be a wasteful process because we actually are losing CO2 rather than incorporating CO2 into a 4-carbon or a 5-carbon molecule. And this process requires energy to convert the 2 of these molecules into a 3-carbon molecule. And so this is a wasteful process. But for some reason, this enzyme will do this under certain conditions. Instead of adding CO2, it will add oxygen. And so what are those environmental conditions that favor photorespiration? And by the way, this process of adding an oxygen is called photorespiration. And you can see why. We're using light energy, but instead of using that to add CO2, we are adding oxygen and giving off carbon dioxide. So this is like a respiration process, giving off CO2. So photorespiration is what we call this process. And so what environmental conditions favor photorespiration? And that is your next question.